and honor to welcome you all to the Molecular Genetics and Biotechnology Seminars 2024, organized by the Department of Genetics, Faculty of Agriculture, Asyut University at Egypt from the 15th of April 2024 to the 1st of July 2024. Today we are gathered to hear a talk about the role of terpene and terpenoid biosynthetic genes from medicinal plants and modulates rhizobia interaction and modulation in soybean. The talk will be given by Dr. Mohammed Ali Abdul Hamid. Dr. Mohammed Ali Abdul Hamid is an associate professor of group genetics and breeding at the Desert Research Center. He received his PhD in 2018 from Agriculture University in China. And now he's working on plant biotechnology in the field of plant functional genomics and secondary metabolites for about 16 years. Dr. Mohammed published many peer papers in top scientific journals like DNA Research, Scientific Reports, Microbial Pathogens, and PLOS ONE, and Frontier in Plant Science, and Journal of Genetic Engineering and Biotechnology. Now, Dr. Mohammed, you can start your talk now, and we will open the discussion after your very interesting talk. I'm sure it will be very interesting. And thank you very much for your kind accepting your invitation in the MGBS seminars. You can start now.
Thank you, Dr. Amir, for this is introduced. And great thanks for my dear friend, Professor Ahmed Salam, for this kind invitation and his team. And thank you to Dr. Abdullah and Dr. Amir. Bismillah ar-Rahman ar-Rahim. Title of our presentation today about the role of terpene and terpenoid biosynthesis genes from medicinal plant modulation, rhizobia interaction, and modulation in soybean glycine max. Will be full screen. Is clear now, Dr. Amir? Full screen?
Yeah, it's clear. Yeah.
Okay, thank you. Before we start our literature today, let us start with a brief introduction about terpene. Terpene or terpenoid are considered from the largest group of natural products and the classes of secondary metabolites found in a lot of plants and many other organism species with more than 40,000 different structures. The terpene recording at first time by Walsh and modified by Razik in 1935. And the first name of this is coming from the Terpensa tree, Basidica terpensa tree. Okay. Terpene have various different classes or different type of terpenoid. For example, monoterpene, which monoterpenoids, which consist from 2 isoprene units, sesquiterpene, which consists from 3 isoprene units, diterpene, which consists from 4 isoprene unit, cistraterpene, which consists from 5 terpene unit, tetraterpenoid, which consists from 6 isoprene unit, tetraterpenoid, which consists from 8 isoprene unit. Just 15 years ago, just many scientists thinking about the secondary metabolism. This is no function, just kind of product by plant, and this is plant used for defense, for perfuming, for attaching some insect, for pollination, something like this. But no one until the revolution of transcriptomic and the metabolomic and the proteomics tools that will already show to us this is secondary metabolism, especially terpenoid and flavonoid, have many important roles in plant physiology. Okay. And let us start with 2 pathways. I think this is 2 focal point or key, master key about pathways that we used in our research today. The first pathway about terpenoid backbone biosynthesis. This is show us this interaction between terpenoid and as a secondary metabolism and primary metabolism like glycolysis. As you see here, terpenoid and different type of terpenoid coming from Malvi and non-Malvi pathway. And this is 2 pathway, sub-pathway coming from glycolysis. This is as a primary metabolism. And this glycolysis coming from starch biosynthesis pathway. Okay. The second important pathway about terpene and the plant hormone pathway. I'm sure most of you at least read one paper or one article about the hormone and the function of hormone like abscisic acid, estradiol, cytokinin, ghrelin, paracetamol, zeatin, and other hormone. But maybe less of you or a few of your researcher know most of this is hormones coming from terpene pathway, especially mulvi and non-mulvi pathway, as you see here. Okay, so this is the focal point about our work. Okay. The idea of our research today coming from when we start our last two our previous two paper, first paper about microbial pathogenesis. This is article we already started the effect about estradiol promoter and its role for promoting rhizobial interaction and increased duration in soybean glycine max. In this article, we just start to look out two genes. This is two gene max1 and max4, and this is two gene related about estradiol, which related about production or formation about hairy root. This is hairy root, the active site for attaching about rhizobium, and this is can generated in the next step to form about or to produce about nodulation structure, okay, or nodule structure, okay. In this paper, we already used some tools like in vitro hairy root, transgenic hairy root, and in this paper, we already look out this is two gene. This is mixed silence for this two gene, and as you see here, when comparing with gas as a control, you see the gas have so much hairy root, and our root and the look out of these two genes just look like smooth or have a small amount of hairy root. Okay. The other inoculum, this is hairy root, as you see here, the effect about knockout. This is no hairy root or less about hairy root. This is no binding site for rhizobium to attach and to form about nodulation. And can you see here about the size and the number of nodules in knockout compare with gas as a control. Okay. The second paper, in this paper, we already try to reverse this point by studies the effect about overexpression about max 4 and the max 1 and the max 3 and the max 4 also. Okay, but this is give us high number of hairy root, okay, comparing with gas, as you see here about slicing about in the figures about use, as you see here, so many hairy root. Okay, this is give us the point or idea to study about terpenoid. Why we study, as we mentioned before, about the terpenoid and the plant hormones. So estradiol, when we start analysis, found this estradiol considered as a terpenoid. Okay, so but not just not just normal terpenoid. This is kind of terpenoid and have so many effects. Okay. So we start to in two article to start to cloning so many genes related about terpenes from Glycine max as a modules and as a model plant, and from medicinal plant, two medicinal plant, Salvia guantatica and Salvia officinalis, and we start to cloning these genes and transfer these genes by using in vitro transgenic hairy root. Okay. The second paper also use the same techniques, but this is at this times we use the successful to cloning genes related about terpene from Salvia officinalis. Okay. We started this work from the beginning. This is work to let me to explain to you how to start this work or the steps that we already used to get successful work like this. Okay. First, we just determination about this sequencing from our transcriptome data, and then make alignment using phytosome 13 database to search about the alignment genes that already found in Glycine max genomics. So why we use this step? This is very important because this is meaning this is substrate of related about genes that already found or recording in this plant. If this is substrate for this gene not recording or not found in this plant, this gene will be transferred without no meaning. They were searching about substrate. Okay. The second, we already do about phylogenetic tree to study the genetic relationship between these genes before cloning and our gene and Glycine max. The third step is that already study about mutative gene expression from phytosome to select. Okay, this is genes prediction, the prediction of these genes and the behavior of these genes, about what is the expression level and what is tissue highly expressed for the genes. As we see here, this most of our gene highly expressed in both leaves, root, hairy root, nodules, stem, flowers. And also we use the other database we call about bar database. This is very important to show us when we successfully to transfer the genes. And okay, so this is genes were highly expressed in which tissue. Okay, so as you see here, most of the genes highly expressed in boots and shoot apical meristem and the leaf and the root. Okay, and the nodules and the root tip. Okay, and the nodule, hair root, and the nodules. Okay, this is the other genes from other plants, Salvia officinalis and Salvia gurantatica. And also use this is database to predict the mutative subcellular localization of our terpene genes based on our adoptive subcellular localization to predict and to understand what is the highly expressed of these genes, which gene was highly expressed in which organelles. Okay, so this is give us so much meaning. Okay. And from the result, we found this mutative subcellular localization. Most of our gene highly expressed in cytosol and the mitochondria and the nucleus and the plasma and the cytosol, which meaning this is when you search about the terpene gene, about Malvi and the non-Malvi pathway, you will find most of the genes already highly expressed in the blastoid and cytosol. Okay. And also we do mutative subcellular localization for gene related, which are related about hairy root and signaling pathway and the nodulation, and we found most of signals pathway and the hairy root and the nodulation pathway also were expressed in the same organelles. Okay, we do this is mutative expression for MAX1, MAX2, MAX3, NFR5, NFR1, MAX4, DMI1, DMI2, DMI3, NIN, and NSB2. And if from the same result, we found the same organelles also. Most of these genes also highly expressed in cytosol, mitochondria, blastoid, and plasma membrane. Okay, after that, we do our schema for our transformation and the prediction. So we follow this step. Okay, the first step, we successfully to cloning our gene, but this is from this schema from from Glycine soja. This is wild type. And the same schema we used for cloning gene from Salvia officinalis, Salvia gurantatica, and from Glycine max. After that, we transfer these genes to in vitro hairy root of Glycine max by Agrobacterium rhizogenus strain K599. And after that, we study the effect of each gene about the phenotypics, about plant, about hairy root, and about the shoot tip. And after that, we start uniculum, the transgenic hairy root by Rhizobium bacterium. We use the strain USDA 110. And after that, we record some phenotypic data like in vitro root length, fresh root weight, nodule numbers, fresh nodule weight. Okay, and calculate. After that, we do GC mass to analysis about terpenoid, but in this case, as you saw here, we use HPLC. Okay. So this step coming, let me explain a step with more details. Okay, we just collect seeds about soybean. This is make sure this is seed was good, healthy, and not have any kind of scratching and not have inside infection. After that, we put all the seed in one layer in the petri dishes, and then put this petri dishes inside glasses big beaker, and inside this beaker have inverted small beaker like as you see here, and we added mix from HCl and NaOH. This will be produced some steams, and this steam will be used to sterilize this seed. And at the top of this is beaker glasses of beaker, we cover by plastic and leave it in the hood, under the hood for 12 hours. And day or after 12 hour already finished, we just remove the plastic and then use collect the seed and washing by distilled water for 3 times, each time for 10 minutes. After that, we start to grow the seed in MS media, B5 media, okay, and then incubate for 1 week in dark. Okay, after that, this is seedling will growing like this, as you see. Okay, after that, we collect this is part from the seedling and cut the really root. Okay, and they keep the cotyledon with a small piece from the really root, as you see here about cycle from red cycle, as you see, also. After that, we use this is cotyledon and incubated this cotyledon with agrobacterium that have our target plasmid for each genes, and then checking for 10 minutes for and added some drops from some chemical acetylseringon to induce the transformation processes plasmid from bacteria to plant tissue. After that, washing about 3 times by distilled water. After that, we prepare some artificial soil. This is consists from perlite and vermiculite, okay, and then we deep about this is cotyledon inside, and the cover this is plastic box by plastic bag, okay, and keeping for maybe 20 to 25 days. After that, we get plant like this, and this is plant have transgenic in vitro transgenic hero, okay, as you see. This is phenotypics about our transgenic plant for 25 days. As you see here, this is not really root. This is in transgenic hairy root. Okay. As you see here, this is after 40 days. Okay. After that, we start to inocule. This is transgenic hairy root by our rhizobacterium. As you see here, if you look here, if you look here, you will found. Sorry. This is remaining part from the really root, okay, and all of this is root. This is all transgenic hair root, and this is nodules that already formed, okay, and this is small nodules, okay, transgenic hair root. After that, we collect this is all the nodules from all the replicate for each gene, and we start to study account this is nodule how many nodules and the size of these nodules, and we take about fresh weight and dry weight of this is nodules. After that, we see the hair root and with more details. We're using about Olympics. This is microscope. This is have good resolution to see the binding site about the nodulation and how many nodules in each 1 centimeter. Okay, the other thing, we make slides about nodules to see inside the nodules about the maturation processing inside the nodules. And this is all our transgenic plant. This is root and shoot phenotypics after 25 days about glycine max. As you see here, each gene have different effect about shoot and about the root, about the plant compared with gas. As you see here, gas have a small root and small shoot. Okay, comparing with the other transgenic treatment. Okay, for example, here we start to transfer gene FDBS genes. This is responsible for Terpene and the other gene from glycine max, GGBBS, glycinide pyrophosphatase. This is responsible for mother component for diterpenes, and the phenol. This is monoterpene, saponin, TBS 6 node, and franzol linol genes, GBS linol, and other linols. After that, we take some record some phenotypic informations like root length, fresh root weight, fresh nodule weight, nodule numbers, as you see after 10 and 20 days. And also we check our gene expression and all our under the effect of our gene transferred by our genes. And this is after 10 days from inoculum by Rhizobium bacteria. As you see here, this is have different about the nodule size, number of nodules, also about the inside of nodules. Also, you see this is go to maturation faster or slowly to go to the maturation processing inside the nodules, as you see. and this is after 20 days. Okay, you see so much nodules already formation, and you see also most of nodules inside go to fast to or quickly to maturation processing. And after that, we collect all our replicate and start about the transition kilo route and started to analysis about terpene concentration and the different type of terpenes using GC mass. As you see here, this is a variation about the concentration and type of terpenes. And we do 6-way Venn diagram to see how much component already common component and unique component under the effect of each treatment or each gene transfer. After that, we collect about hair root after 10 days and nodules after 10 days, and also nodules and hair root after 20 days, and check the effect about each gene from our terpene genes on the different gene which related about nodulation and nodulation signals and about hair root formation. This is nodules after 10 days, and this expression of our target genes and the effect about nodulation genes in hair root after 20 days. And this is effect about overexpression of our genes, terpene genes, on the hair root and the nodulation genes after 20 days in nodules. After that, at the final, we start to drawing reconstructive mapping. This is from the first time, from the first look, you see this is look like simply busway, but this is complicated busway. This is coming from combining about more than 16 busway together. Okay, and this is show us the link between our genes and the nodulation and stragolactose pathway. For example, this is Malvi and the non-Malvi pathway, as I see here. This is GBBS. This is mother component for monoterpenes, and this is DMB, ABB. This is go through this is sub-pathway to zeatin, and zeatin use the cytokinin, and this is the teams to follow. This is effect about CRE1 and the LHK1 cytokinin signals, and this is the end product of this is effect about cell division and nodule formation. Okay, and if you look about GBBS, this is go through this is FBBS and FADBS genes, okay, to forming about ghrelin granule pyrophosphatase, and this is FBBS and FADBS. This is from soybean and from Salvia granulatica, effect about parasinosteroid biosynthesis, and this is effect about VR1. And this is used as a parasinosteroid single, and the final effect about this is signals cell elongation and cell division. Okay, and if you look about the other pathway about granule pyrophosphatase, this is have three branching. The first branch is about terpene, and the second about ghrelin biosynthesis pathway, which effect about ghrelin signals, and this is effect inside the nucleus, and this effect about transcription factor in ACB1 and ACB2, and effect about nin also as a transcription factor. And this is effect about many gene expressions. This is genes related about nodulation formation. And the other branching about carotenoid pathway, this is to create or forming about MAX1 and MAX3 and MAX4, and this is stragolactose biosynthesis. This is the effect about auxin, and the effect about recollaboration, and the effect also about abscisic acid, jasmonic acid, which effect about calcium spiking and calcium ion transfer, and as you see here. Okay. Thank you.
Thank you very much, Doctor. And we now can open the discussion. If anyone have any question, you can unmute your mic and ask the question directly, or you can write your question in the comment, and we can read it for Dr. Mohammed. Thank you for this very interesting talk, actually.
Thank you, Mr. Amr.
So I have a question, small questions. Maybe I didn't get it at the beginning. So these genes are not presented in soybean. So So the plant itself, it doesn't have this, it doesn't contain these genes, or you overexpress them all?
We actually we already cloning two genes from soybeans. This is FVBS and GBBS. Okay, and on the other side we cloning other gene from two plant about Silvia officinalis and Silvia grantacrea. This is
But they are
yeah,
not
yeah.
present in soybean.
This is not sorry. This is not found in this plant, but they have alignment genes already. So
Ah.
the question now about why we use this gene, and this is genes from Silvia grantacrea or Silvia officinalis, give us more effect or high efficiency compared with as you see here in this figure. Okay, why give us more efficiency compared with these two genes from Glycine max? Okay, this is secret behind this is about the type of the type of domain and motif found in these genes. Okay, if you see about this is you see these slides.
Yeah.
Okay, if you see here the gene of from Silvia and Silvia officinalis, Silvia grantacrea have very important traits. Okay, why? This is have different kind of domain and motif inside one gene. Okay, on the other hand, about genes of terpenes from Silvia from Glycine max maybe have a few number of motif and domain. Okay, so this is give us flexibility or give us how to say and I'd say this is can if one gene have so many domains and the motif inside of this domain gives this gene ability to produce so many kind of terpenes compared with other genes. Okay. So
Okay.
if you so if your analysis takes these genes from Silvia and compare the genes with Glycine max, okay, you found the same sequence, okay, but these genes from Silvia or from medicinal plant have so many motifs and have so many domains. Okay, for example, these genes, as you see, have about 4, and in some cases, this is we have about eight domains. Sorry, 8 motifs inside the genes. So each domain and each motif give availability to the genes for product for produce so many components compared with the other gene from Glycine max. It's clear?
Yeah, yeah. Thank you very much.
Yeah.
Do we have any more questions? I think there is no more questions. Okay, thank you, Dr. Muhammad, for this very interesting. Yeah.
And bioinformatics, another question?
Yeah, there's one more question from Abdullah.
Yes, can
So
bioinformatics be employed to predict the extent of gene in plant if a primary gene coding the hormones found in bacteria region to plant using gene technology? Yes, Dr. Abdullah. But depending about how many and the kind of bioinformatic tools that you already used. Okay, so as you see here, we do when we start our experimental, or this is data of this article. We use so many bioinformatic database. Okay, we start about dry lab, as you say, or in vitro lab. Okay, before or in silico lab or dry lab as you call, or before any experimental. So we detect these genes. We start to detect the substrate of these genes in the plant. Okay, and when we start to cloning this gene, okay, which kind of domain and motif in this is in the sequence that we already cloned. Okay, and if I start to transfer these genes, what is the effect about these genes? The end effect. So we use the bar database to see, oh, this gene will be highly expressed in root, in nodules, in boots, as you see. Okay. After that, genes will give you how many products. Okay. And this is genes when I started to include the transgenic route with bacteria, with rhizobium. Okay, what is the effect? So before we do the experimental, let me tell you, we predict more than about 80, more than or higher than 800 80% from this is work. They will be successful, and this is data we get by bioinformatic tools. Okay, so this is depending about how many tools, which kind of gene that we already select, and which database I will be used. Okay, to predict this. It's clear, Dr. Abdullah? Dr. Abdullah?
Yeah, I think it's clear. Thanks.
Yeah. Yes, thank you. Okay.
Thank you, Dr. Mohammed, for this very interesting talk. And thank for all the audience for this, for having here in the MGBS seminars. And we wish that we can see you again for more seminars.
Thank you, Dr. Yeah, thank you, Dr. Irfan. Thank you very much. Thank you.
Thank you.
Yeah.
Goodbye.
Goodbye.
Bye.